So where's sort of all of this come from? Around 5 years ago, there was the 13th International Forum of Ovarian Cancer, and a number of strategies were discussed in terms of how we can improve our earlier diagnosis of ovarian cancer and also improve ovarian cancer prevention measures. Some of the things that were highlighted were the use of the contraceptive pill, which can reduce the risk of ovarian cancer, and we'll talk about that shortly in a bit more detail. Another focus was repurposing of drugs with low toxicity profiles as preventative agents, as well as looking at surgical measures such as self-injectomy, so removal of the fallopian tubes versus removal of the ovaries, or the oophorectomies, or a combination of the two, to see whether we can reduce ovarian cancer through these measures. And then another large focus of the think tank group was to improve our genetic testing of relatives of affected women, and also to look at population testing for specific genetic mutations in higher risk groups, particularly BRCA mutations and mutations in other genes called the mismatch repair genes that's associated with a syndrome called Lynch syndrome. So what do we know about ovarian cancer risk? Whenever we're talking about risk of any disease, there's factors that we call non-modifiable, things that we can't change, and other risk factors that we call modifiable, so things perhaps in our lifestyle that we can change. In terms of ovarian cancer, age, so there's an increased risk of ovarian cancer, as there are with most other cancers as we get older. Family history increases the risk a little, and then genetics. So a lot of people will have heard of the Brachy gene. So mutations in the Brachy gene is rare. Well under 1% of the population will have a mutation in the Brachy gene, but there are some ethnic groups in whom there is an increased risk of being a mutation carrier, particularly Ashkenazi Jewish population, where there's somewhere between a 4 and 6% incidence of mutations in the Brachy gene. And there are a number of other groups of populations where there's an increased incidence of Brachy mutation. There are also abnormalities in what we call single nucleotide polymorphisms, and I'll talk about those in a minute, but they are things that can be found more commonly on our genome, and that can also be associated with an increased risk of ovarian cancer. There's hormonal factors, such as how many children or pregnancies a lady's had, age at menarche, age at first birth, and age at last birth, as well as age at menopause, can increase the risk of ovarian cancer. And that seems to be linked to the number of ovulations a lady has in her lifetime. And then certain inflammatory conditions, like pelvic inflammatory disease and endometriosis, can be linked with an increased risk of certain types of cancer. Some of the modifiable risk factors are things like diet, smoking and exercise, and also hormonal factors, particularly contraceptive pill use. So a number of studies that are published have shown that use of the contraceptive pill can significantly reduce the risk of ovarian cancer. We know that if a lady takes the contraceptive pill, so the combined oral contraceptive pill, so that's estrogen and progesterone containing pill, for 10 years, that can reduce the risk of ovarian cancer in relative terms by around 50%. And we believe that that benefit is sustained throughout a lady's lifetime, even if the contraceptive pill is stopped. So probably in terms of medications that we know of that can reduce the risk of ovarian cancer, combined oral contraceptive pill is definitely the most significant. So what are the objectives of the Progress trial? The first is to better understand public and GP perception of genetic testing and of population risk assessment. The second objective is to test the feasibility of a cancer risk algorithm that we've designed. And this algorithm encompasses what we call polygenic risk scores, which I'll talk a little bit about in a minute, and also epidemiological data. So that's information about family history, parity, contraceptive use, and everything. And it aims to guide the personalization of ovarian and breast cancer screening and prevention in the general population. And we also aim to determine whether these algorithms can be used in conjunction with population BRCA screening to help further define risk in certain populations. So the background behind the trial, we know that genetic risk assessment has the potential to improve the impact and efficiency of current prevention and screening programs. But although there's a national screening program for breast cancer, there's no ovarian cancer screening program. And we believe that the identification of people who are at higher risk of ovarian cancer may improve survival benefit associated with current screening modalities. But at the moment, there's really very little consensus on who should take prevention measures, what they should do, at what age, and how long for. So just to talk a little about single nucleotide polymorphisms. So as you probably know, our DNA contains all our genetic information, and it's essentially made up of letters A, T, C, and G. And within the population, there are variability in the letters at certain sites on the genome. And a single nucleotide polymorphism is when there's a change that we see in over 5% of the population. And that's different to a mutation. For example, a Brachy mutation, which is an abnormality that's seen in less than 1% of the population. This is all talking about changes in the sequence of the letters on our genome. And we've found that there are over 300 sites on our genome where variability in the letter can be associated with an increased risk of breast cancer. And now actually nearly 100 spots or sites on the genome where variability in the letter can be associated with an increased risk of ovarian cancer. Now, if someone has a change or a variant in just one of those sites, the risk increase will be tiny. But if we look in a person at the variability at all the sites, so all the 120 odd sites that have been linked to ovarian cancer, and now there are over 300 sites that have been linked to breast cancer, and we look at the variability on all of those sites, we can start to calculate someone's SNP risk, or what we call polygenic risk score for a certain disease. And that can be used for SNPs that are associated with other diseases as well, such as heart disease, and Alzheimer's, diabetes, and others. And it's these single nucleotide polymorphisms that we're looking at in this study that have been linked to breast and ovarian cancer to calculate what's called a polygenic risk score. So we know that polygenic risk scores are powerful tools to further define specific risk profiles. A good example of this is the fact that even in patients with BRCA mutation, the polygenic risk score can affect someone's risk of breast or ovarian cancer. So we know in women where there's a mutation in the BRCA gene, their risk of breast cancer in their lifetime can be as high as around 60% on average. But if that woman, as well as having a BRCA2 mutation, has a large variability in the single nucleotide polymorphism associated with breast cancer, i.e. a high polygenic risk score, then their risk of breast cancer can be as high as 90%, the top line. But those with a low polygenic risk score, with a BRCA mutation but no other abnormalities in the breast-associated single nucleotide polymorphism, their risk of breast cancer in their lifetime can be as low as 40%. So we know that by combining tests for BRCA mutation and also looking at SNPs to determine polygenic risk scores, we can further refine a lady's risk of breast or ovarian cancer. To date, no studies have used polygenic risk scores in guiding ovarian and breast cancer screening prevention strategies. And we think it's important that we screen women for breast and ovarian cancer risk together to allow us a balanced approach in terms of prevention measures. For example, we know the contraceptive pill reduces ovarian cancer risk significantly, but can also increase the risk of breast cancer. So by further defining a lady's risk of ovarian cancer and breast cancer, that can help decide how we balance risk prevention measures, such as the contraceptive pill use. The PROGRESS trial has two aims. The first is to assess public and GP receptiveness to genetic testing and personalised screening programme. So this part of the study aims to recruit 5,000 participants. These are men and women over the age of 18 on the West London primary care database, and they're simply asked to fill in an online questionnaire that asks lots of questions about knowledge of genetic risk and risk factors, and how people feel about potentially having a risk assessment for cancer or other diseases performed. And the main impact is to determine perceptions of genetic screening and to determine what people feel a level of risk might warrant prevention. The second part of the study aims to conduct a personalised ovarian and breast cancer risk evaluation in women in the general population using our risk algorithm that we've designed. We aim to recruit 500 participants, and these are women aged between 30 and 60 who would be attending their GP practice for any reason at all, or in fact women who approach us directly because they've heard of the study. The study involves taking a saliva sample, and that saliva sample gets sent off for genotyping so we can look at single nucleotide polymorphisms and determine someone's polygenic risk score. The second part of this bit of the study is that we ask participants to fill in a questionnaire that allows us to capture epidemiological information. So that's questions around contraceptive pill use, parity, family history, and other epidemiological factors. And the third thing we do is we take a blood sample, and that is to allow us to take a BRCA test for not everyone in the study, but for women in the study in whom we feel there's over a 10% chance of them having a mutation in the BRCA gene, and we calculate that through various well-established methods. We then take all that information and we calculate the participant's risk of breast and ovarian cancer, and around 6 weeks later we meet with the participant again and communicate that risk score with them, and I'll show you the algorithm that we use in a minute and how that looks. We then will talk about what the risk might be and what measures someone might take to be able to reduce that risk or potentially undergo screening to help catch any potential cancer at an earlier stage. And the study also offers genetic counselling to participants if they wish. We then follow participants up six months later to see really what impact the risk assessment has had on lifestyle, whether they've made any changes to lifestyle or any prevention behaviour that may have occurred because of the knowledge of risk of breast and ovarian cancer. So this is just a schematic diagram of how the algorithm looks once we've put the information in. So you can see here on the graph there's 3 lines. The blue line at the bottom is a person's risk of ovarian cancer by age. So absolute risk on this side, age on the bottom axis, and this is the red line is the risk calculation that we've undertaken for this particular participant. And you can see that with this participant, the risk of ovarian cancer is significantly higher than the average age, so the red line compared to the blue line. And what we can see is on the green line, we can adjust that person's risk if they've taken the contraceptive pill. So we can say to a lady, if you take the contraceptive pill, your risk will have reduced from, say, 0.89% to 0.75% through the use of the contraceptive. The other important use of this algorithm is to look at what age we might recommend screening for breast cancer, for example. So in this country, we offer screening for breast screening to women from the age of 50 for the majority. The reason that the age of 50 has been selected is that we know that at the age of 50, the risk to a woman of developing breast cancer is around 2.5%. And the government has decided that they will offer breast screening to women once their risk reaches 2.5%. Now, in some other countries, they've selected a slightly lower risk of, say, 1%, and they will screen women from the age of 40. But in this country, it's 50 because that's when a woman's risk reaches 2.5%. Now, some women with a strong family history of breast cancer, or those with known mutations in genes such as the Brachy gene, can be offered screening from an earlier age, a younger age of usually around 30, because we know that those groups of women, their risk of breast cancer reaches 2.5% at a much younger age. And what the risk calculation that we use here in terms of breast cancer risk, it can tell us at what age a woman's risk reaches 2.5%. So for example, here you can see on the blue line at the bottom, this is the average risk. When we look at 50, age of 50, the absolute risk is 2.5%. This red line here is the breast cancer risk that we've calculated using our algorithm for this particular participant. And you can see that her risk reaches 2.5, her risk of breast cancer reaches 2.5% around the age of 43. So what we can do using this algorithm is by using genetic and epidemiological information for an individual person, we can determine what age their risk reaches the threshold risk for screening. And there will be lots of women who, using this method, we calculate their risk reaching 2.5% well under the age of 50, and a large number of women whose risk won't reach 2.5% until well over the age of 50. And we think that this method will help streamline screening and make it more cost effective, because we'll be screening those women at younger age who need screening, but we won't necessarily be screening those at older age until their risk reaches 2.5%. So the PROGRESS trial is a collaborative study between myself and James Flanagan. We also work very closely with the genetics counsellors and with the Imperial NAHR team. So that's the National Institute of Health Research. And it's a combined venture between ourselves in the oncology department and the primary care team. And we think that's really important because ultimately a lot of risk assessment will be done in the primary care setting. We're also working in conjunction with Sophia Genetics, so that's the company that are now owners of the algorithm that we designed, between myself and David Cox. And a number of other people as well, including the GP Consortium and the OCAT committee as well. So what do we expect to get out of the study? Well, we feel this is the first study to evaluate the behavioural effects of population genetic risk screening using a combined risk algorithm. We feel it will provide us with pilot data for further studies on population genetic screening. And we feel it will help us to power and design nationwide trials comparing a personalised approach to selecting women for screening versus the current standard age selecting process. So where's the trial up to at the moment? So part A of the study, the questionnaire study, opened just getting on for 2 years ago now, January 2019. And we've recruited just over 5,000 participants to the questionnaire study. So we're now just starting to look at those data, and we hope to analyse them and write up our results in the next few months. Part B of the study, which is the more interventional part of the study, where we take saliva samples and convey risk scores to patients, that opened actually in January of this year. It recruited really well initially. We recruited just over 80 participants between January and February. And obviously lockdown came along and the study was suspended. It's just reopened this last week for recruitment, and we've started to recruit patients again. So we're hoping that's going to pick up again pretty quickly. That's all the slides I've got. Happy to answer any questions.
Thank you very much, Jon. That was a really great insight into your work and, you know, the algorithm. And I really like how you'll be following up with participants 6 months after that initial risk score to see, you know, their view on the approach and if they've made any changes to their lifestyle as a result. So thank you very much. So I've got a few questions that we were sent in beforehand, but if anyone watching has any questions for Jon, please pop them in the Q&A or the chat and we can ask him now. So firstly, to kick us off, how do you predict women will respond to having their risk score calculated, and can you foresee any challenges around asking women to make lifestyle changes?
It's a good question. It's something I've not touched on really in these slides, but you know, the psychological and emotional impact of having any sort of risk assessment performed is something that's really important to this study. And we actually we asked participants to complete questionnaires at the start of the study, also when we've given them their sort of risk assessment, and also 6 months later. And it's a very sort of standardized, I suppose, a mental health sort of psychological questionnaire to help us assess the psychological impact of such a discussion. And you know, so far, I would say that you sort of see two sorts of reactions to it. One is very much a feeling of empowerment and a positive sort of response to it, that someone becomes more aware of their potential risk and feels more empowered about things they might be able to do to reduce that risk. And you know, obviously for others there is some anxiety that can be provoked from discussions about risk, and you know, that's something that's important to capture, because we do feel that this is going to be a very impactful type approach, but that people will need to be supported well through it, as people often are with any sort of genetic counselling or risk assessment.
Great, thank you. And this is not only working with members of the public, it's also working with GPs, and one of the aims of the trial is to help GPs discuss risk and prevention with their patients more effectively. And what do you think is the key to getting GPs on board to adapting to a new way of working? Because I can imagine they must be bombarded with guidance for all different diseases all the time.
Sure, I think it's a good question. That is a big focus of the study, is to engage with primary care and the GPs, because ultimately our vision for the future is that it will be in the primary care setting that these sort of risk assessments are performed, and not just for cancer risk, but for risk of other diseases as well, with high blood pressure, diabetes, and heart disease, for example. And that's already being done by GPs in the community, you know, in terms of blood pressure control and prescription of drugs like statins to help reduce cardiovascular risk in those deemed to be at higher risk. So it's all about education, really, and ensuring that the people and like the GPs undertaking the risk assessments understand what it is that we're doing, how the algorithm's designed, and what measures people can undertake to reduce the risk, or where risk reduction is not possible, what screening measures could be employed to help detect cancers earlier.
Great, thank you, John. And then someone's asked, why does the cancer come back so quickly? That's quite a general question, but can you give some insight on that?
I guess the question is probably asking about ovarian cancer.
Yes, yeah.
Cancer doesn't always come back. We know that, unfortunately, in around 70 to 80% of patients diagnosed with ovarian cancer, there is a recurrence at some point. And we feel that the main reason for that is that we're still not good enough at detecting ovarian cancer early enough. But a lot of women diagnosed with ovarian cancer are diagnosed after a cancer cell has already escaped from the ovary or the fallopian tube. And although surgery and chemotherapy can be very effective at treating that, and in some cases curing it, in a significant proportion of women, a cell sort of manages to resist the surgery and the treatment, and at some point starts to grow again. And it's partly or predominantly due to the stage at which we're diagnosing cancer, ovarian cancer, which is why we feel that employing screening and risk and prevention measures for ovarian cancer is so important.
Great, thank you. And someone has asked, or kind of, my mum died 7 months ago at 72 from what I'm told was a rare aggressive form of ovarian cancer called Carcinosarcoma. I'm waiting to hear if I'm eligible for BRCA testing. Is this cancer type linked to BRCA mutation?
So we know that women diagnosed with ovarian cancer, or the commoner types of ovarian cancer, will have a mutation in the BRCA gene. That means 80 to 85% won't. But the majority of women still don't, but we know ovarian cancer flags up a person's potential chance of having a BRCA mutation, because we know the chance can be as high as 15 to 20%. Carcinosarcomas would be included in that group of ovarian cancers where there is a higher chance of there being a mutation in the BRCA gene than average, but it's still only around a 15 to 20% chance.
Great, thank you. And someone else has asked, how does somatic BRCA fit with research, and how relevant is it to prognosis in terms of germline BRCA?
Okay, so a somatic BRCA mutation means the finding of a mutation in the BRCA gene in the tumour. Now, if we find a mutation in the BRCA gene in the tumour, that could be because of one of two reasons. One, that person was born with a mutation in the BRCA gene, and therefore that mutation is in every cell of the body, including that ovarian cell that has then gone on to become an ovarian cancer cell. The second possibility is that a person who develops ovarian cancer was not born with a mutation in the BRCA gene, and the BRCA gene within that cell that became a cancer cell was normal, but during the process of that ovarian cell becoming an ovarian cancer cell, an abnormality occurred within the BRCA gene. And we know that that happens in around 5% of women with ovarian cancer. So around about 5% of women with ovarian cancer will have been born with a normal BRCA gene, but the BRCA gene in the ovarian cancer cell has become abnormal during the process of the cell becoming a cancer cell. And so if we find a mutation in the tumour, in the BRCA gene, we would offer that lady germline testing. So testing their blood or their saliva to look at their non-cancer cells, normal cells that they were born with, to see whether there's a mutation in the gene in the normal cell. If there is, that means that they were born with the abnormality, and that's why we're seeing it in the tumour. If we don't find a germline BRCA mutation in the blood or the saliva, but we find it in the tumour, that means that lady's not been born with the abnormality in the gene, but they developed it. Now, that's not hereditary. That's not something they can pass on. But it is relevant in terms of the that person's cancer being more likely to be sensitive to drugs like carboplatin and PARP inhibitors. So that's not to say there's no BRCA mutation, the cancer is not responsive to those drugs. We just know that if there is a mutation in the BRCA gene in the tumour, then there's a higher likelihood of benefit from drugs like platinum and PARP inhibitors.
Great, thank you. And someone's asked a really interesting question. Do you see a future point where, for example, PARP inhibitors would be given as prevention if a woman is BRCA positive and high PRS and family history is devastating?
Yeah, so again, really good question. Unfortunately, we don't know the answer to that question. And there's lots of discussion around clinical trials of drugs like PARP inhibitors in women who are at higher risk of ovarian cancer. PARP inhibitors are relatively well tolerated drugs for most people, but they are drugs that can have side effects. And the difficulty with contemplating the use of drugs like PARP inhibitors as a preventative measure is trying to weigh up the potential side effects of the drug without knowing perhaps what benefit you might be inducing by using it. So at the moment, PARP inhibitors aren't used for that reason. There may well be some studies in the future looking at that.
What are the common side effects of PARP inhibitors?
So the commonest side effects are things such as tiredness.
Yeah.
Make you feel more tired. They can cause some sickness, but we can normally help prevent sickness with anti-sickness medication. And they can disturb sleep sometimes. Those probably are the three commonest side effects, and they tend to improve with time. So we know that when a lady first starts a PARP inhibitor, those side effects can be more prominent, but over the coming weeks or months, they can reduce. Other side effects are things like muscle aches and pains, joint aches and pains, and the drugs can also lower immunity a little bit, not to the same degree as chemotherapy, but they can lower the white blood cells. They're the commonest side effects.
Yeah, so I totally get what you're saying. You know, the risk might be high, but you know, to suffer those side effects for a long time just might not be worth it. And then we've just had one more question, which is quite similar to one I asked before, but someone has asked, would potential recurrences carry the same somatic BRCA? What about difference between BRCA1 and 2 somatic?
So the answer to the first question is not necessarily. So occasionally we can see what we call a reversion. The cancer cell, there could be a mutation in the BRCA gene, and over time a further mutation occurs within that same gene that almost corrects the initial abnormality that occurred. So the reason mutations can have an effect is that a gene encodes a protein, and that protein has a function within the cell, and the role of BRCA within the cell is to repair damage to DNA. And if there's an abnormality in that gene, the cell's not able to repair damage so well, and that either helps to promote the development of cancer or stimulate or help the growth of that cancer cell. But mutations can occur again so that that initial mutation gets corrected and the structure and the function of the protein that the gene codes for is normal again. And that's one of the ways that we think cells can become less sensitive to PARP inhibitors. And that probably happens, as far as we know at the moment, in around 2% of women with a mutation in the gene, in the tumor.
We've just had a question in, and they're asking, what is the process of BRCA testing?
What's the process of BRCA testing?
Yeah.
So that would slightly depend on the context. So certainly my clinics and a lot of the clinics in this country, in terms of ovarian cancer clinics, there's a process called mainstreaming, which is where the oncologist looking after a lady with ovarian cancer can actually offer and undertake the blood test for BRCA testing within the cancer clinic, and then send that blood test off to the regional genetic centres, or the genomic centres now, to get the test done. So any woman in this country with ovarian cancer is eligible for BRCA testing for the commonest types of ovarian cancer. In someone who's not got ovarian cancer, or perhaps never had any cancer, the BRCA testing on the National Health Service is undertaken through the regional genetic centres. So every area of the country will have a regional genetic centre. So for example, in Northwest London, it's at Northwick Park Hospital. And a GP or another doctor looking after someone can refer that person to the regional genetic centre. They will then get sent a questionnaire to fill out things like family history. That gets sent back, and then the geneticists at the regional genetic centre will determine whether that person meets the criteria for the genetic test, like BRCA testing. And that's how it works on the NHS. Privately, people can also pay for BRCA testing.
Yeah.
Either privately through going to see a geneticist. There are also ways of ordering a test online without even speaking to a doctor, and you know that's slightly more controversial, but people do it. So there are companies that will offer counselling online before a genetic test is done.
Great, thank you so much. And I think I can't see any more questions coming in, so I think we'll end the questions there. But yeah, thank you so much for joining our session today. We really appreciate your interest and support. And a big thank you to Jon for taking the time this evening. You know, I'm sure you're really tired after a really busy day at work. You can find more sessions to join on the Staying Connected part of our website, including patient support groups. Thanks again for joining us today, and I hope you all have a lovely rest of the week, and I look forward to seeing you at our next research webinar. So yeah, thank you everyone, and to Jon.
Thank you.
Thank you.